Welcome everyone to Australian Biocommons AI in the Life Sciences webinar series. My name is Georgia. I'm the Australian Biocommons BioCloud Training and Communication Officer, and I will be your host for today. Artificial intelligence is rapidly changing the way we do science. This webinar series will help you understand how AI is being used in life sciences right now in real-world research. You'll hear directly from experts working across biology, medicine, agriculture, and the environment as they share how AI is helping them and their research. Before we begin, we'd like to take a moment to acknowledge the traditional owners and their custodianships of the land on which we meet today. In my case, this is the Gadigal people. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connection to country. We recognize their valuable contributions to Australian and global society. Today, we're thrilled to welcome Dr. Anna Trigos to open this webinar series and inspire all of you at the beginning of your own AI journey. Anna is a group leader in the computational biology program at Peter Mac. She leads the Multi-Omic Evolution and Ecosystems Lab, focusing on understanding cancer evolution and ecosystems through the analysis of omics data and the application of computational methods. Anna developed the first spatial-based gene expression signature in prostate cancer during her postdoc and built and led her team to develop novel bioinformatics tools to analyze microenvironments. In 2020, she was awarded the Lee Award Medal, recognizing emerging female research leaders by the Peter Mac. And in 2021, she received the Young Investigator Award by the Prostate Cancer Foundation. She's been awarded funding from NHMRC and the CAS Foundation as lead CI and has received collaborative funding from the Prostate Cancer Foundation and the Department of Defense in the US. Welcome to the webinar, Anna.
I thought first I'd give a little bit of background on me and kind of my educational background, a bit of my lab, and how and why we started to doing AI and a bit the journey for us. So a bit about me first. Yes, you have a bit of a starting point. So I'm originally from Venezuela. I did my undergraduate in biology, which would be very similar to a degree in undergraduate degree here in biomedicine. And I did an honors year in cellular neuroscience. There was very little, there was no bioinformatics really. There was a little bit of R, but that was about it. So as a original training, I'm a biologist. I then came to Australia and I did my Master of Science in Bioinformatics at the University of Melbourne. And then I did my PhD in Cancer Bioinformatics at Peter Mac through the University of Melbourne. And during my PhD, um, I worked with many types of data, but primarily bulk RNA-seq. I also looked at SNPs and copy number data from DNA sequencing. I used primarily publicly available data from the Cancer Genome Atlas. I also did a bit of network analysis and some pipeline development during my PhD. So you would say it's a very much of a traditional kind of PhD project, so to speak. Then in 2019, 2020, I started working with spatial proteomics data. This was very early on, but we were looking at opal multiplex immunohistochemistry, which was just 7 or 8 markers, and we continue analyzing this type of data now. At this point, I started collaborating with clinicians and not just with wet lab scientists. And this is the advantage of being based at the Peter Mac because it's a research institute and a hospital integrated under the one roof. In 2021, I started working with single-cell RNA-seq, multi-omic DNA-seq. So basically, let's say I was bioinformatician moving with the times, getting access to new data types, developing methods, analyzing data. We were also generating data at this point. I started my lab in end of 2023, so we've been a little bit over a year already. We primarily work right now when it comes to the lab as an entity. We focus on understanding treatment resistance and response from a cancer evolution and ecosystems perspective. And for this, we use a lot of omics data, genomics, transcriptomics, epigenomics, single cell, you name it, we use it. And we also started a bit on patient imaging, where the AI kind of story comes in. In the group, the background of people are bioinformatics, computational biology, engineering, math, stats inclined people. We have someone with a background in deep learning. We also have a clinician and an endocrinologist PhD student. My lab is completely a dry lab. So we have access to the lab, so we don't do any pipetting or anything like this. 100% a dry lab. We do have our own research questions and also develop our own bioinformatics methods. And we primarily work in multiple myeloma and prostate cancer, but we apply our methods to many other cancers as well. And we have a number of collaborations. So one thing that I realized, especially when I started in my lab, was that bioinformatics in the classic sense, and when I say bioinformatics, I'm talking about analysis of some sort of omics data. Um, I realized that there were many limitations of what you could do. So, for example, it's largely tissue-based. So all the analysis of omics data, right, comes from some sort of tissue, some sort of cancer tissue, normal tissue. And this really is only capturing only part of the phenotype of the disease that we are interested in. So it's only a very small part of the story, what is happening in the omics. The other frustrating thing is that for the vast majority of projects, you have samples, in a single location at a single time point in a patient, and we know that, cancer can disseminate across the entire body, can change over time, etc., etc. This is true for many other diseases as well. So then the question is, we spend so much time looking so deeply at individual samples or even large cohorts of samples, but it makes you wonder all about all these things that we're actually missing. And then there's the issue around how do we translate then findings to patients. So for example, if you have a project and you find something that you think might be a target for a new treatment, then you need all this extensive wet lab validation that then happens outside of kind of the realm of your lab. If you find something that could be a biomarker, then incorporation of biomarkers into the clinic is actually quite tricky. You have to have very large clinical trials, international trials, etc., etc. Um, so this sounds very negative. I'm still very passionate about bioinformatics, and we do quite a bit of bioinformatics in the lab, and most of the people in my lab do bioinformatics, will continue doing it. But I think there's still this a little bit of frustration of a little bit the limits of what we can achieve with bioinformatics if our goal is to have a very big impact on patients. So that kind of got me thinking a little bit of on the AI space. So AI means, I know it's a very vague term. So I know many people really don't like just using the term AI. I'll be using it here with a very specific definition, in this context. Um, so I want to make a few distinctions, and then we'll get to what I'm talking about. So there's AI that is used to make your life easier, right? So those are the co-pilots and the ChatGPT, etc., etc. So I'm not talking about that. Yeah. And then there's let's say AI as new algorithms. So what do I mean by this? These are the new algorithms that are being developed for the analysis of omics data. A good example of that is in, for example, spatial transcriptomics, where you have a lot of data, you have the whole transcriptome, and you have single cell resolution, etc., etc. And many methods using AI, deep learning, computer vision, something like this, are being developed for things like clustering or cell type identification, etc., etc. So that is a very that type of AI, it's a little bit of maybe like an extension of bioinformatics, new tools that we can use as bioinformaticians to analyze our data. And that is not necessarily the topic of today's talk and what I mean when I talk about AI and bringing AI into the lab. What I'm talking about is about AI that expands the type of data that I can look at beyond what I can do with traditional bioinformatics. So with traditional bioinformatics, I'm kind of stuck with omics data in a way. But if I want to look at patient imaging, if I want to correlate with patient outcome, if I want to build prediction models, if I want to do etc., etc., at a very high level beyond your basic random forest and linear regression. And that is what I'm talking about when I talk about AI in this talk. I'm bringing AI into the into our research projects. So what do we want to do? So a particular interest of ours is looking at patient imaging. There are many types of patient imaging. We're particularly interested in PET scans and CT scans for, for various reasons that kind of go beyond the scope of this talk. And the idea is to combine the information contained in patient imaging with multi-omics data to better understand disease phenotypes and better predict patient response to treatment for better personalized medicine, right? And we do this, we have projects across different cancers with different clinicians, and I'll talk a bit about this in a second. But this is roughly the goal and what we want to kind of achieve to be doing long term. So from bioinformatics to AI, so I try to draw a little bit of a path of the sort of things that we have encountered, we have had to think about, a bit of the unexpected things, so that hopefully others after us can learn from our experience. So for me, if you're thinking about AI and large cohorts and the sort of things, then you have to think about what sort of collaborations you need. Unless you are a clinician who is also a deep learning person, etc., you probably need to work with someone who can give you access to patient data. Or you can get obviously publicly available one, but I'm talking here about if you want to start something from the ground up. Then you have to think about funding. Where's the money going to come from? Then you have to think about the patients. What sort of data? What sort of cohorts? What sort of and all the intricacies about that. Then there's the data, getting the data, then that's a bottleneck. Then, okay, you have the data, staff and students, how do you get them? How do you incorporate them into your lab? Then you have the issue around compute. How do you then analyze this data? Then what? You do the research, you get amazing results, and then what do you do with what you have achieved? So these topics is what I'm going to be covering in the next few minutes for the rest of my talk. So the first one is collaborations and funding. So if you are interested in applying AI in let's say new cohorts for very specific let's say clinical question or clinical gap, you probably have to start thinking about large cohort studies or getting access to standard of care samples. And standard of care samples are those samples or data that are obtained as part of routine care of patients. Yeah, so a patient goes to a hospital and gets tested and these sort of things. Normally, the good thing is that I think there is a very big effort for in the clinical space to put together these large cohort studies, to put together these large registries, these large clinical trials, to collect samples, to collect data. So there's already a lot of effort I find in Australia in these things happening. So you probably don't have to start from scratch. You could probably just start collaborating with someone who has access to these large cohort studies. It's important also in these collaborations to try to define what problem you're trying to solve. And this is research 101. But I think when we think about prediction models and sort of things, we often think about survival, progression, pathology classification. But what we have learned is that these are often not necessarily the only or the most interesting questions, clinical questions that are out there, or clinical gaps. Sometimes it's a matter of disease identification. Where is the tumor? How much tumor there is? Even those sort of questions can be of great interest and of great value to address. So don't think that necessarily your research needs to end up with some sort of survival curve prediction outcome kind of thing. When it comes to funding, the good things about these collaborations, then you have access to funding that more traditional funding like through NHMRC, for example. But then you can also do things like MRFF if you have clinicians and you have implementation scientists, etc., etc. So there's certainly a lot of opportunities to join the sort of big big efforts, and I think the funding climate certainly currently supports that. So okay, so you've got the collaborations and you've got the funding, great. Now it comes to the patients. So I mean, obviously you need to have proper consent for the patients from the patients to use their data for research. And normally when that translates as is basically a lot of paperwork. So you have to think about the ethics applications, about the governance applications. You know, are you piggybacking over projects that are already there? Are you starting something from scratch? So it's a lot of meetings and a lot of paperwork that need to happen in this space. One challenge here is that um, often not many people are that well versed with what doing research in this space looks like. Think about it like, you know, let's say bioinformatics 10 years ago when people really didn't know what it was about or why you just couldn't use Excel. So it's a little bit like this. It's like, okay, AI, well, you just use R. People know R and Python now. Um, but I think an understanding of you know prediction models about well statistics is not yeah there's no power you know what I mean, all of these things need to be demystified and make it a bit more it changes completely the sort of problems and caveats and things that you need to think about and communicating these to people, you know, ethic review committees, the governance people, to the clinicians themselves, etc., writing it in patient consent form, all of these things is actually quite can be quite tricky and quite time consuming, but obviously critically important. Then there's another consideration here, which is around the idea of bias and in AI, and the most commonly thought of one is around ethnicity and race, right? So we know that if you're looking at very large data sets, depending on the type of data, you are potentially able, you know, there are batch effects, so to speak, based on precious ethnicity and race. So then the question is, you know, what do you do about that? No, do you have, how do you consider that in the research that you're doing? Do you have to consider it in the research that you're doing? Um, if you want to include it, then does the data exist? All right. Does this information exist somewhere? Do you know, this patient is of this background, or do you not know this? If you don't know it, then how do you get this information? And if you do know it, is information reliable or is it self-reported? So this is just one kind of tiny example of the sort of things that we start asking, that you know committees people might start asking, and it's very hard to give good answers for. Our go-to answer is, well, it's a pilot. We'll see how we go. But you know that cannot be the answer forever. And certainly there are many other biases that you could potentially think of in the data, right? From where it was collected, you know, how much pollution there is in the area, lifestyle factors, etc., etc. And you know, people say the more data, the better, because like that the signal, you know, becomes stronger, etc. But my intuition has always been that the more data, the noisier the data, because there's much structure in the data, and then that makes it tricky to use down the track. How do you address these issues? We do not know. I do not know, but I think the field will eventually sort it out, because I do think it will become a problem if we're thinking about, you know, applying these methods in the clinic or in another setting. Then, okay, so you've sorted out all the paperwork, you've got the data, right? You've got the samples. Now, it's a lot of logistics to get access to the samples, to generate the data. Again, a lot of paperwork. You have to talk with pathologists, you have to interact with the core facility managers. That data generation, so here I'm talking about some sort of omics data generation, you know, it would be similar to the sort of problems that we had, let's say, as a community bioinformaticians later, it's a similar set of issues. I guess the difference here is that often there isn't a wet lab biologist in the mix, and it's basically you are the clinician, and there is no wet lab scientist, so then that makes data generation something that then you really need to think about. Um, so that it changes a bit, very much dynamic of how that needs to happen. Then the second point is around the logistics to find and identify data. So clinical data, patient imaging data, pathos results data, etc., etc. You have to think about, this data is in patient medical records. Who is going to access it? Are you going to access it? Do you have the ethics to do it? Does our clinician has to do it? Do you have to hire someone to do it? There's a lot of considerations there. In our experience, to kind of access the data, access the samples, access all of this, we have had to interact with people that I never thought I would have to interact with. So things like nurses, clinical trial coordinators, medical physicists, anatomical pathologists, etc., etc. So it's really, when you say multidisciplinary, this is really a multidisciplinary effort to get all of this data available for you. So once you get the data, okay, you got the data, you've got a folder with the data, you're very happy now, and now you have to think about who's going to do the work. Um, so in general, there are kind of two ways to go about it. You can have bioinformaticians who are learning AI on the job, and I think that is probably easier if you have projects where you are kind of so you have spatial transcriptomics data, you want to apply some sort of AI tool onto it, and then you kind of try it out and such. It's kind of very familiar. But if you're looking at different types of data, new types of data, that can take quite a long time. So you're fortunate enough to be able to hire someone with a deep learning background to help us in this. If that's possible, that is something to consider. But I think having good linkages with the universities, with the computer science departments, I think that is critical. In my experience, there's a lot of linkages between, let's say, bioinformaticians and biomedical research or biology departments. That linkages are very strong, but there is much less so of bioinformaticians with the computer science department as is there's less so with the statistics and maths department. This is just my impression. And I think building those are very important. It actually has been quite tricky for us to do that, and that is something that we're still working on. Then it comes, so you've sorted out the people, you've got the data, and then you have to think about the compute. Where is the work going to be carried out? So in our experience, high performance compute systems that were, let's say, designed for genomics might not work for AI. They might not have the GPUs, they might not have the stability, they might not have enough cores, they might not have enough, etc., etc., etc. And that has been our experience. The good thing is that in Australia we have POSI and NCI, which are a very large high performance compute that are available for researchers to access. And there's obviously also cloud computing. Then, okay, you have sorted out, okay, I can use one of these systems to carry out my research. That's amazing. But then you have to think about, okay, can we transfer the data from where we are at the institute to these systems? Who do we need to ask permission to do this? Then you get questions, okay, who owns the data? And then when you think about it, is do I own the data? Does the clinician own the data? Does the institute own the data? It becomes very murky very quickly. Depends on your institute, the policies and the governance around that might be more developed, more mature, or it might not be. So that takes quite a bit of work. A good thing we have things like, for example, NCMAS, which is a grant scheme. I learned about it just last year. It's a grant scheme that allows you to request compute access to POSI and NCI to get significant amount of compute, and that really so you will get the compute for free. So at least you don't have to worry about the funding side of it. A challenge, though, that we have found in using these systems is that you really need very advanced HPC skills. So although we are dry lab and everyone has a some sort of bioinformatics background, some people have even done masters of bioinformatics, etc. The level of HPC skills that are needed to work in these systems are often more advanced than what we would need if we were to use the HP systems at for example, University of Melbourne, or we were to use the ones internally at Peter Mac. So it has taken quite a bit of upskilling of people in the computational skills to be able to use the systems, to be able to do the research. Yeah. Okay, so you've gone, you have the data, you have the people, you have the compute, you do the research. And science is great, so you get amazing results very quickly, and it's all fantastic. And that's great. Congratulations! And you publish, you write your paper, and you publish your paper. And I know this slide is summarized in like 5 years, but I guess I didn't want to get too much into this because I think we all know the challenges with science. So okay, so we publish the research. That's great. But then if you think about how do we then translate it to the clinic? Yeah. How do we then take what we have learned, whatever model, whatever system, whatever classification, whatever we have done, how do we get it from that to actually having an impact in the real world, right? So this is a very evolving landscape in Australia, but I'll give kind of a super brief overview. So normally, people are very comfortable with the idea of not comfortable, but there is a path of translation of, a drug to the clinic. You do a clinical trial, you do this, you do that, you contact pharma. There is some path. With AI, the path is not that clear. So an AI tool is a product. Yes, it's not a drug, obviously, it's a product. And it falls under the category of a medical test. So it's kind of similar to, so let's say when you go to the doctor, you have a blood test and measure, iron levels, and you get a little number in your document. That is, through equipment, through a machine, yes, that spits out this number. Well, AI kind of fits under that medical test category. Um, the challenge, the unique challenge here is that AI, whatever product you have, is a model that has been trained on patient data. So it's kind of not independent of the data that you have used to generate the model. So I'm not talking about the generalizability of the model and all of these things, and that's the science part of it. I'm talking more about, can you separate the product from the patient data, and then if not, is then who owns the product and how does that, if the data comes from multiple institutes, etc., etc., can get quite tricky. And this is new territory in Australia. The legislation is not that developed. So we in our group are not nowhere near this yet. We're just starting to do AI at the moment, but we are having these conversations. And the reason for that is because one concern of mine is that were there any considerations that we should be thinking about when we are designing our research projects, when we're thinking about the patient cohort, the type of where the data comes from, how is data processed, etc., that we need to think about to enable the translation of whatever we develop in the end. And there are no clear answers for that at the moment. I think it's still a bit of an open question, but it's something to keep in mind and keep on top of, because the last thing you want is to do all of this effort, get something that works, and then for a logistical reason, then you cannot really implement it. And so just two final, very general conclusions, take home messages for this. The first one is that, extending, let's say, bioinformatics research to include AI, under my definition of AI, is not just about learning, new methods. It's not about, oh, and let's use PyTorch and TensorFlow and let's learn how to do that. It's really about the need to build an ecosystem. Things look different. The types of people you interact with are different. And when it comes to the collaborations, what we've noticed is that we now collaborate more with clinicians rather than biologists, because the big data comes from clinicians, and the big data from big cohorts comes from clinicians rather than biologists, which have more of a kind of case control experimental design. That is for the sort of research that we want to do. That is our experience. And finally, just a big thank you to my lab, to my clinical collaborators, many other people who have helped the research that I didn't put here, but this is just the ones that have are kind of relevant to the talk I've given, and the funding agencies as well. Thanks.
Thank you, Anna. That was great.
Thanks.
So we have time for questions now. If you want to ask Anna a question, you can use the Q&A function on the Zoom dashboard. Anna, you mentioned that it's very important that there is this collaboration with bioinformaticians and computer science professionals.
Mhm.
So you've managed to do this successfully, it seems. So how do you think we can improve this kind of connection and collaboration?
Oh yes, look, it's tricky, and I would say it's a work in progress for us. Um I think things that would definitely help is um having a way of having a bit of more maybe visibility to our projects and our research in these departments. So I would love to have, for example, things like invited talks, shared symposiums, shared seminars, this sort of things to just foster these things a little bit more naturally. I think adding a bit more I think internally, I think advocating for the importance of these interactions is critical as well. I think the I think for the, let's say, average biologist, the thinking is that, oh, the bioinformaticians can just do AI as well. And I think they don't get that that is actually quite different and you need a very different set of skills. So I think advocating for that message internally in our institutes is important as well. I think having bioinformatics degrees or computer science degrees that are a little bit less one or the other and having them a little bit more fluid and more open would allow that the students that come next a bit more trained and a bit more open-minded, and like that over time you would create linkages. So I think it's a work in progress. I don't think there's a good formula, um, but yeah, I think things will definitely improve over time.
You also mentioned the need for compute, getting the infrastructure, and how do you manage to communicate its value to decision makers, control funnels,
Oh,
research
that
institutes?
that that is a work in progress as well. It's quite tricky. I think I think goes back to this wanting to really extend this message around how um AI, deep learning, computer vision is just so different to traditional bioinformatics, and therefore the computer needs are different. I think that message is critical and needs and that's the message that needs to get through. I think also, getting access to the external computer and actually showing their power and their ability, and showing what can be done with them. I think that is a very important part of the puzzle. You know, eventually everybody would have to be doing some sort of AI, right? Just like a few years ago, we couldn't imagine what lab scientists doing in bioinformatics, but now they are is becoming more common. The tide will flow, and then AI will become part of that. And I think at that point, probably we'll start getting more support for compute. But I think there's certainly avenues that one can explore currently.
How close we are to automate the research pipeline, so from data collection to model prediction.
Yeah, so I think there are a lot of efforts to try to streamline this. I think it will take quite a few more quite a few more years to get all the cogs running quite well. I think there's a lot of will and desire and a lot of interest in this and how this could be applied. Um, so I don't think it will take that long because there is a lot of will, but I think eventually we will eventually get there. I would put it maybe 4 or 5 years there would be less pain points and more streamlined path.
Yeah, it sounds like, I mean, it is that we are still everyone's learning, right? We're more or
Yeah.
less all on the same boat. And it's, part of what with Biocommons we do is, we
Hmm.
try to make the national, scale digital infrastructure more accessible and
Hmm.
usable. And now we have to integrate all of these new, AI skills and tools that we need. Do you have any advice on what are the key skills that people need to use AI on the HPC? Do you know if there are places where we can learn? How are you dealing with this?
Yeah. So what we're doing is, as yeah, someone has commented there, NCI and Policy has very good training on how to use the systems, and it's training that is designed for people who know how to code, are very comfortable with a language, and they want to use HPC at advanced skills. There are quite a lot of training there available, and a lot of the material is available online. One thing that we have really had to focus on is, for example, building containers. Before we didn't used to containerize stuff, now we have to containerize everything, so that's one thing. The other thing is, for example, data management. Sometimes um, if you use an external compute, you cannot just leave the data there forever, so you have to really have a strategy to manage that. Um, so I think it's a combination of computational skills and data management skills to make sure that you can actually use these HP systems at the maximum without costing an arm and a leg. Yeah, and doing testing and benchmarking of tools and this kind of rigor in the computational analysis that I think often bioinformaticians don't worry so much about it. If the script is working, that's it. It's great. But now the amount of compute that you're using is that much, then you have to really think about how to optimize the script. How much compute do you actually need? How much time do you actually need? You really have to work on that skills quite a lot.
What's the best way to choose the right analysis for huge amount of data, such as genomics, proteomics, or transcriptomics? So we focus specifically on the data we need without being distracted by all the data that is irrelevant or just noise.
Yeah. Look, that is a very tricky question to answer. I think it really depends on what your goal is, what your research question is. You know, I mentioned there previously in one of my slides around you have more data, or more data times is not always good. You have to really know what type of information you need, and what is not useful, you have to discard. I agree that there's this thinking of, chuck everything, put everything there, the more the better. But I think it needs to be, clean data. It needs to be the relevant data. You don't want to have, redundant data. The more redundant data, the more noise you have. So I think thinking around that is certainly critical when you're doing these types of big data research.
Yeah. So we have to leave it here for today. Thank you so much, Anna. This was amazing. You can find out more about the upcoming webinars, workshops, and other events on our website, and watch recording of these and previous events on our YouTube channel. And finally, we'd like to acknowledge our funding. The Australian Biocommons is enabled by and created by Bioplatforms Australia. Thanks so much for watching. We hope you enjoyed today's presentation. Thank you, Anna, and we look forward to seeing you again soon. Until next time, goodbye for now.